Thank you. I'm happy to do so. So I'm happy to kick off this first presentation series of our keynotes block 1, which is psychedelics and mental health. Our first speaker is Dr. Katrin Preller. Her scientific background is clinical psychology. She did her PhD on the neurobiological long-term effects of cocaine, MDMA, and heroin use. As part of her postdoc, she investigated the effect of psychedelics on self-perception and social cognition with works abroad at the UCL and Yale University, where she also holds a position as assistant visiting professor. She got appointed a junior group leader at UZH, where she's working on the cognitive and emotional processes in health and disease, and using multi-modal neuroimaging techniques. Her work is well received and respected, which is reflected by her receiving the Pfizer Research Award and the Swiss Society for Biological Psychiatry Young Investigators Award. Dr. Preller is a member of the Mind Foundation Scientific Advisory Board and will also give a talk at this year's Insight Conference in September. Her talk for today will be the neurobiological mechanisms of psychedelics and implications for psychedelic-assisted therapy. So I'm really happy for your talk right now. I'm looking forward to, and for all the listeners, feel free to post your questions in the Q&A section below.
Thanks a lot, Christoph, for the very nice introduction. Let me share my screen real quickly before I get started. Okay. Good. So, yeah. So first of all, thank you very, very much for the invitation. I'm really glad to be able to open up this wonderful symposium that the organizers have put together. Thank you so much for that. I'm very much looking forward to the other talks which follow today, which I'm sure will be really, really exciting. So for this opening talk, I will talk a little bit more about the basics of psychedelics, about how they work in the brain. I'm going to present results that we have of studies that we have conducted at University of Zurich, mostly with healthy participants. We are conducting studies in clinical populations as well, and there will be a few talks throughout the day which will present these results. So here I'm going to focus more on the mechanistic effects of psychedelics, but I will also at least try to show you how I think that these effects relate to psychedelic-assisted therapy and why it is important to understand the neurobiology of psychedelics if we want to leverage them for psychedelic-assisted therapy. So, as you all know, psychedelics have a lot of effects on how we perceive ourselves and how we perceive the world. But of course, we also have these increasing amount of clinical trials looking at therapeutic effects of psychedelics with so far pretty promising results. Now, to be able to conduct this psychedelic-assisted therapy in an optimal way, I think that we really need to understand what these substances are doing. And of course, since we're working mainly in the field of psychiatric disorders, it's a good plan to start looking at the brain if we want to understand what they're doing and why they can be helpful. So there are a lot of hypotheses for why these substances could be helpful. And I'm just going to present you a few of them. It's a comprehensive analysis of why they could be helpful would probably not fit on one slide. So we can imagine that these substances change the way serotonin to A receptors are expressed in the brain, and there are first studies looking at that. We've also seen in animal studies that psychedelics induce neuroplasticity. There are a couple of studies looking at neuroinflammation. There are other studies looking at epigenetics. Then we see alterations in brain networks that might be helpful for patients. We also see alterations in reward and emotion processing. A lot of patients report increased insight in dysfunctional behavior. And we see an increase in social connectedness. Now, 30 minutes will definitely not be enough time to walk through all these potential mechanisms. So I'm going to focus on 3 of them: alterations in brain networks, alterations in reward and emotion processing, and increased social connectedness. And I'm trying to make the link of why I think that these changes that we see in the brain and in the brain interacting with its environment might be helpful for patients. And I'm also trying to talk about how we can leverage these effects and how we should take them into account when we're conducting psychedelic-assisted therapy. So the psychedelics, the 2 substances I've worked most with are psilocybin and LSD. They induce pretty similar effects, but the pharmacology is a little bit different. So psilocybin is a preferential serotonin 2A and 1A receptor agonist. LSD, on the other hand, is a little bit more of a promiscuous drug. It stimulates various serotonin and dopamine receptors. But the effects of these substances, as I mentioned before, are pretty similar. So I'm showing you these 2 plots because they will pop up again and again, probably, and not only in my talk, but also in the talks that will follow later today. So both substances induce an altered state of consciousness that is characterized, first of all, by alterations in our visual sensory experience. So how we see the world. That's why they've also been called hallucinogens. However, these are not the only effects, and that's also the reason why we prefer to talk about psychedelics now, because they also induce changes in how we perceive ourselves and how we perceive our body, how we feel connected with our environment. Now, as I already mentioned, when we were talking about the neurobiology of psychedelic substances, a good place to start is not only the brain, but it's a specific part of the brain, which are the receptors. So, as I said, psilocybin and LSD have a somewhat dissimilar pharmacology, but they both stimulate the so-called serotonin 2A receptor. And we wanted to find out how the serotonin 2A receptor contributes to these effects that our participants are experiencing and are reporting. And what you can see here on this slide are 2 studies, one with psilocybin, one with LSD. And in both studies, we blocked the serotonin 2A receptor with catenzerin. So what catenzerin does is it blocks it's an antagonist, so it blocks the serotonin 2A receptor. So when we administer the psychedelic afterwards, it can stimulate all these other receptors that it's targeting, but not the serotonin 2A receptor. And in both cases, you see that if we administer catenzerin first, it's the green bars over here and the yellow bars under LSD, we reduce these subjective effects to a degree where they're not distinguishable from placebo anymore. So for both substances, we need the serotonin 2A receptor to have these psychedelic effects and experiences. So psychedelics, as we know now, enter the brain, they stimulate the serotonin 2A receptor. But how do we get from the serotonin 2A receptor to these distinct altered state of consciousness that our participants are experiencing? And how do we get from there to these therapeutic effects that we are observing in clinical trials? Well, first of all, what happens after a psychedelic docks to the serotonin 2A receptor is that it changes how active these neurons in the brain are. Since the serotonin 2A receptor is an excitatory receptor, it increases the likelihood with which these neurons in the brain fire, the likelihood with which they transmit information. Now, of course, these neurons are all connected in a network that is the brain. So if these neurons fire differently, That means that the brain is connected differently, that it transmits information from one brain area to the other in a different way. And of course, our brain is not an island. So our brain interacts with our body and we interact with our environment. So having a different information processing within the brain obviously will have implications for how we perceive ourselves and how we perceive the environment. Now, let's take a first look into these networks and how information is being processed while we are under the influence of a psychedelic. So to start off, I want to give you a bit of an introduction to the anatomy of the brain. I'm sure a lot of people in the audience will be familiar with that, but it's really good to keep this picture in mind. So there is something which we call a cortical hierarchy. We have sensory brain areas, and these are the colored brain areas in the brain. So they receive input from within our body and from our environment. So for example, and I'm just pointing that out because it will come up a couple of times, the pink areas here are responsible for receiving visual input. And the other brain areas here are, for example, responsible for auditory input, for touch, so basically for all our sensory modalities. Now, of course, we don't perceive the world as just a visual impression or just an auditory impression. We need to connect these different sensory experiences with each other, and also, of course, with the memories we have, or with planning what we want to do next. And to integrate all this information, we need these gray brain areas, which are our association brain areas. So we don't only have the cortex in the brain, we have subcortical structures as well. And one particularly important subcortical structure is the thalamus, so this big blob here in the middle of our brain. And it has been hypothesized that psychedelics act by reducing the filtering function of the thalamus and sending more information to the cortex, causing a sensory overload that is then experienced as psychedelic symptoms. So, as I said, the thalamus is usually filtering out important from non-important information that we receive from within ourselves and the environment. And in our studies, we wanted to test this hypothesis if the filtering function of this brain structure in the middle of our brain is indeed reduced under the influence of a psychedelic. So what we did here with participants under the influence of LSD is we conducted an analysis which is called functional seed-based connectivity. So we looked at how the thalamus is connected to the rest of the brain, how much in sync it is with the rest of the brain. Now, keeping this picture that I showed before in mind, you see this differentiation between these sensory brain areas, so the ones which were colored, which were the colored areas, and these blue brain areas, which are our association cortex. So what that means is that the warm colors in this slide, as well as in the ones following, they are more strongly connected to the thalamus, whereas these blue brain areas are less connected to the thalamus under the influence of the psychedelic. So indeed, it seems like the thalamus is more connected to these sensory brain regions. However, as always, all these neuroimaging techniques, as well as any other scientific method we use, has limitations. And in this particular case, we cannot talk about causation. We cannot talk about directionality. So we just know that the sensory brain regions are more in sync with the thalamus, but we don't know if the thalamus is indeed sending more information to the cortex, because it could be the other way around too. So to be able to test this particular hypothesis, we had to use another method, which is called dynamic causal modeling. And this method does give us directionality between brain areas. However, of course, this one also comes with disadvantages. And the disadvantage here is that we, at least for the time being, cannot look at the whole brain. but we need to restrict ourselves to specific brain regions. And of course, to test our model, we restricted ourselves to brain regions which we had an hypothesis about because of the thalamic filter model. And what we see here is, again, red arrows are increased connectivity and blue arrows are reduced connectivity. So we do see indeed that the thalamus is sending more information to specific brain areas, and it is also sending less information to other brain areas. So basically replicating what we've seen in our functional connectivity analysis already. However, in contrast to this original thalamic filter model, it doesn't seem like the cortex is just overflown by information, like it's like there is too much information and it's just everywhere in the cortex. It rather seems like the thalamus is sending more information to specific brain regions. And as we've seen in this previous slide, it seems to be mainly these sensory brain regions. Now, again, just a reminder, keep this cortical hierarchy in mind also for the next slides to come. Colored areas, sensory brain regions, white areas in that case, association brain regions. So obviously, after we have established that there is a reduced filter function, that there is more information flow to the cortex, we also wanted to know what's going on with the cortex, with the rest of the brain after the thalamus has sent more information there. And there we used another method, which is called global brain connectivity. And global brain connectivity gives you the connectivity between each voxel of the brain with the rest of the brain. And what you can see here really nicely again is this differentiation between these sensory brain networks and these association brain regions. And the other thing that you can see nicely here is that we basically get exactly the same effects, whether we are using LSD or psilocybin. Now, what we see here again, the warm colors increase connectivity. So we have an increased connectivity between the sensory brain regions, increased sensory processing, which is not necessarily surprising given that we also have this reduced filter function and this increased connectivity from the thalamus to these brain regions. But we also see this decreased connectivity in association brain regions. So the frontal cortex, for example, here. And what that means is probably that that our brain is integrating this information that we are receiving from our environment and from within ourselves in a different way than it usually does, because we see this decreased connectivity in these association brain regions. So to sum this up, we have seen increased connectivity from the thalamus to sensory brain regions. So increased sensory processing, which is not counterbalanced by our association capabilities. And what that means is that we probably integrate the information we are receiving in a different way than we do usually. And that can, on the one hand, explain things like why we have these visual alterations under the influence of a psychedelic. But it can also explain why people under the influence of psychedelics report to see the world and themselves in a very new way, in a new light. And this, this breaking of rigorous thinking patterns, this perceiving yourselves and the environment in a different way, could help patients to break out of these rigorous thinking patterns that they usually suffer from. For example, in depression, this ruminative negative thoughts that a lot of these patients are suffering from. So that is one mechanism by which psychedelics could help psychiatric patients. I want to quickly go full circle with this part of the talk by again talking about receptors. So we have not only blocked the serotonin 2A receptor and looked at the subjective effects that psychedelics are inducing, we also looked at what ketanserin does to these neural effects. And what I'm showing you here is LSD versus placebo, so the picture you've already seen. And underneath, we see LSD versus a condition where we block the serotonin 2A receptor and then gave LSD. And as you can see, just by eye, they're looking very, very similar. And the reason why I'm not showing you placebo versus ketanserin plus LSD is because it's a really boring picture. It's basically a gray brain. So we don't see any significant changes in connectivity when we block the serotonin 2A receptor and then administer LSD. So not only is the serotonin 2A receptor responsible for the subjective psychedelic effects, but it's also responsible for the neural effects that we are observing when we're using fMRI. We also used another method to validate that, and we looked at the serotonin 2A receptor gene expression in the brain. So where is that receptor expressed? Where is it located in the brain? And as you can see really nicely here, is that the receptor gene expression maps very nicely to the changes in connectivity that we observe under the influence of LSD. So where this receptor is located has something to do with how information transfer changes in the cortex, which makes sense, because obviously if you have more information, or if you have more receptors in a certain brain area, you can assume that it would be differently affected by the psychedelic substance. Now, another thing before I close this part of this talk is that we also looked at how the brain is organized under the influence of placebo, so in its usual waking state. And we looked at how, if there is a correlation between the functional architecture of every individual's brain with how they react to a psychedelic. And indeed, we found a pretty strong correlation here. So it seems like the functional architecture of our brain on an individual basis might be predictive of how we then react to a psychedelic substance. So the second part of my talk is really about having this brain, which is processing information very differently from our usual waking state, and how this relates to how we interact with our environment. So one important finding that has been replicated by now quite a few times is that under the influence of a psychedelic, we are reacting less to negative emotional stimuli. And we see that when we do behavioral tests like here. So the error rate is higher for negative stimuli if we do a computerized test under the influence of psilocybin. And also, again, when we look at participants' brain, we see that the amygdala is not that reactive to negative stimuli. And this is particularly important if we think about depressed individuals who have a very negative bias. So they continue to think about negative experience. They also react more strongly to negative stimuli. And this could be a mechanism where psychedelics normalize this negativity bias we observe in depressed individuals. But of course, negative experiences are not only restricted to showing people negative pictures, but one thing that is happening a lot especially with psychiatric patients, is that they have negative social experiences. In particular, they are often excluded from their social environment. So what we wanted to find out is if psychedelics also influence negative social interaction, and in particular, social exclusion processing. We used a very standard paradigm for that. So what we did is we had our participants coming into the lab. They were introduced to Michael and Sarah, and they met them again while they were lying in the scanner. And in the scanner, Michael and Sarah played a ball game with them. So they could throw so our participant could throw the ball to Michael, Michael could throw the ball to either Sarah or our participant, and so on. But what happened then is that after a while, Michael and Sarah started excluding our participant So they only threw the ball to each other without playing it to our participant and excluding them from the game. And the beauty of that paradigm is that it induces consistently a feeling of social exclusion, even though, you know, when you look at it right now, you might think that, wow, that's not too bad. But basically every single participant coming out of the scanner told us like, wow, that was really, really mean. And the other beauty about this paradigm is that it has been replicated over and over again. So usually what happens if our participants undergo this paradigm is that we see the anterior cingulate cortex lighting up when participants are excluded. This is why this signal in this anterior cingulate cortex has also been termed the social pain signal. And when we do the same thing with psychiatric patients, in this case, borderline personality disorder patients, we see that this signal is even stronger in these patient populations. So what happens when we do the same thing with psilocybin? Well, first of all, we see that the exclusion rating, so how strongly people felt excluded under the influence of psilocybin, was reduced. Obviously, we are working with a highly psychoactive substance. So we asked a lot of control questions, including or trying to find out if they were aware that they were excluded. The dose wasn't too high in that study. So it's not surprising that they were indeed completely aware that they were excluded. So all our control questions didn't show any significant differences from placebo. The only one that did show significant differences was this exclusion rating. So people were aware they were being excluded, but it didn't feel that bad as it does under the influence of placebo. And also when we look at our brain imaging data, we see that the social pain signal in the anterior cingulate cortex is reduced under the influence of psilocybin. So what does all of this now mean for psychedelic therapy? Well, first of all, we have seen that with psychedelics, we change how the brain is processing information. But as I said, the brain is not an island. It is interacting with our environment, and we also perceive our environment very differently. So psychedelic-assisted therapy is and needs to be an interaction between a pharmacological and a non-pharmacological therapy. So the environment that the therapists are providing and the way we are conducting the therapy is important because we do react differently to this environment. So we've seen that these changes in brain connectivity and in information processing might be leading to opening up new perspectives, opening up a new view of the world and of ourselves. So the therapist can certainly support this process of finding new perspectives. But we also need, if we're talking about that, we also need to think about timing. So at the peak of the experience, it will probably be terribly hard for the participant or the patient to interact with the therapist. We've run a few other studies as well, looking at this brain connectivity. And what we've seen there is that also in the beginning of a psychedelic experience, we see these changes in alterations or increased sensory processing and decreased association functional connectivity. So maybe a lower dose might even be already enough for this to happen, to support this finding of new perspectives. And that is something we should test in future studies. And also, of course, the timing is important. So we don't quite know yet how long these effects last and when the best way or when the best time is to engage with the participant in finding these new perspectives. So we've also seen that there is a reduced reaction to negative stimuli. So that might give us or give the therapist the opportunity to engage discussing negative life events with the patient. And again, here we don't really know quite yet when the perfect timing for that is, but we have seen that this reduction in amygdala reactivity for negative stimuli is reduced under the acute influence. We've also seen that it is increased the day after the experience, and then it is reduced again and lasts for about a month. So we need to understand this, this emotional reactivity a little bit better to determine the best timing to discuss negative life events with our participants. And we've also seen that psychedelics change the way we perceive our social environment. And this is something that is going to be important for psychedelic-assisted therapy, because a lot of patients report that they are socially isolated, that they don't have a lot of social support, and are feeling alienated from their social environment. So taking this effect into account, this increase in sociability, maybe this provides room for including, for example, family members or friends in the therapeutic process. And we've also seen that we might be able to find objective biomarkers that the way our brain is organized in our usual state might be predictive of the way we react to a psychedelic. So maybe we have an objective stratifying biomarker for people to determine who will benefit from the therapy or not. Obviously, these measures have so far been only obtained in healthy participants. So we need to replicate them in clinical populations, and this is something we're doing right now in our clinical trials. And with that, I want to thank everyone involved in this research. And from some of these people on the slides, you will hear more later today. And I'm really looking forward to their talks and, of course, the next talk, which is following right now. Thank you so much for your attention.
Thanks a lot, Katrin, for this wonderful talk, for this introductory talk about the neurobiology behind psychedelics. If I have a look on the clock, we have actually time for one question, which fits perfectly for our Q&A section, because we have one, and which is from Anne-Marie, and she asks, as there are a lot of thalamic subdivisions, is there a particular thalamic seat you have chosen for your study in 2018?
Yeah, wonderful question. Thank you so much. We haven't. So right now we have indeed looked at the whole thalamus, because it was the first study to really investigate that. But moving forward, we should definitely parcellate the thalamus according to its nuclei and take a much closer look at the specific role of each thalamic nucleus.
Okay. Thanks a lot.